join the meeting. I see 30 people have already joined. So a couple more minutes and then we'll get going. Thank you. Hi, good morning, everyone. So welcome back to Grand Rounds this morning. Hey, Eric, how are you? I appreciate it. Good morning, everyone. So we've got a special Grand Rounds this morning. We have our own internal Dr. Jane Witt with us this morning. Dr. Witt is an associate professor in cardiovascular medicine and studies the role of endothelial function in solid organ transplant rejection. He has published his research in leading journals of the field, including Nature Immunology, Nature Communications, and PNAS. Dr. Jane Witt received his MD PhD from Case Western Reserve University, his internal medicine residency at Stanford, and then is a former fellow physician from our program at Yale. He has stayed on since been on faculty since 2016. He joined early as an instructor and then an assistant professor, and was recently promoted to associate professor in the section. He is a recipient of multiple awards, including the Internal Medicine's Physician Scientist Award, the Eva Dostanek Award for translational approaches for studying cardiac allograft vasculopathy, and the American College of Cardiology's Young Investigator Award, as well as seed grants from American Medical Association. And from the American Society of Transplantation, he received the Young Innovator Award. We're really excited to have you here, and please join us.
Well, thank you all for attending today, and it's a real pleasure. I know it's an opportunity to speak here today. I'm actually going to take off my mask. That's okay here today. So again, thank you all for coming. Today, I'm going to be talking a little bit about aging syndromes and vascular health. This is kind of a divergence from what our lab typically studies, which is translational approaches for solid organ transplantation. Let's see. The screen here is frozen. Let's see. So these are my disclosures. So it's well appreciated over the past 4 decades that the population, the U.S. population, as well as the global population, has undergone a dramatic shift in demographic, and we've seen a disproportionate increase in the cohort of patients who are above the age of 65. We see here a dramatic increase in both the male and female populations that increase in disproportion to the rest of the population. And it's well appreciated that concurrently, along with this age population, this age population shows an increased predilection or predisposition to the development of cardiovascular conditions. Here shown are many cardiovascular conditions we treat every day: coronary disease, heart failure, SVT, most saliently AFib, and stroke. So not only is the population aging, but the aging population shows an increased predilection for these cardiovascular conditions. And so it's important, or it's becoming important to study the field of aging in order to define a molecular basis for the aging process and attempt to try to reverse this process to prevent a lot of these senescent or age-associated conditions. And this is just to note here that along with the aging population, this has changed our practice, that along with cardiologists, we are simultaneously becoming geriatricians, in that many of the cardiovascular conditions that cardiovascular procedures that we perform today, and these are the invasive ones, but also the invasive and non-invasive medical therapies as well, are geared towards this advanced or aging population. So aging is a very large topic, and it's something that is new for me in my lab. There are different theories associated with aging, and some of them are listed here: toxic metabolic byproducts, oxidative stress, reactive oxygen species, cumulative environmental injury, and genetic predisposition. So today, what I'm going to be discussing is a genetic predisposition towards aging. I'm going to be focusing on a syndrome called Hutchinson-Gilford progeria syndrome. And the reason why I'm focusing on this condition is because among all the progeroid or advanced aging syndromes, Hutchinson-Gilford progeria syndrome, or HGPS, most faithfully recapitulates the hallmarks associated with aging, which I'm going to show here. The clinical manifestations of HGPS very closely mimic those associated with aging in normal individuals. And this condition is the molecular basis of this condition is very highly well developed and studied. HGPS was studied by Francis Collins, who was the former head of the NIH. And so the molecular basis underlying this condition has been very well established, to the point where drugs have been developed to try to reverse aging. And this is really a nice example, an illustrative example of how the molecular basis of a condition can inform a therapy. And I think anyone here said, here, take this pill, can live 10% longer, or undergo this genetic manipulation procedure, live 30–50% longer. This is clearly an unmet need. And it's an ultra-rare condition, so it's a substrate for orphan drug approval. So there's a lot of interest in HGPS as a model for aging. And in the latter half of this talk, I'm going to be discussing a gene that my lab discovered. It's called Z521 that recapitulates a lot of the aging phenotypes associated with HGPS. So Hutchinson-Gilford progeria syndrome, or HGPS, is a genetic disease. It's a monogenic disorder that involves the lamin A gene. I'm going to discuss here some of the clinical features associated with HGPS, as long as a molecular basis of it. Hutchinson-Gilford progeria syndrome was named by Hutchinson-Gilford. These were London surgeons, which in the early 1900s, late 1800s, they described a cohort of patients which developed these aging or advanced aging phenotypes. They called this syndrome progeria, from the Greek meaning early, and then geria for old age, and they termed this condition progeria. HGPS is a sporadic disease. It's inherited as an autosomal dominant in an autosomal dominant fashion with complete penetrance. It's an ultra-rare condition. So this condition here affects between 1 and 4 million children. It occurs sporadically. There's approximately 500 known cases of HGPS in the world today. It's diagnosed principally by a whole exome sequencing, which is a new procedure. There is no ethnic predilection for this disease. There's no male or female predilection for this disease, and there's no risk factor for its inheritance. Typically, children with HGPS undergo a normal perinatal and postnatal development and do not carry a family history of HGPS. In 2003, the molecular basis of HGPS was becoming more defined. There were two groups that independently found that the lamin A gene mutation was causative for HGPS. One group here is from the INSERM, CNRS in France, and the other was by Francis Collins' group at the National Human Genome Research Institute, or NHGRI. Following this discovery here, the molecular basis of how lamin A could cause HGPS has been widely or comprehensively studied by many groups. So lamin A is part of the nuclear envelope. So in all cells and higher order organisms, metazoans, the nucleus is compartmentalized inside a structure, the nuclear envelope. This nuclear envelope is composed of an outer membrane and an inner membrane. And then underneath this inner membrane is a nuclear lamina. The nuclear lamina is composed of intermediate filaments. And these filaments, it's composed of lamin A and lamin C. And these filaments act as kind of a scaffold or a structure, if you will, to allow anchoring of critical proteins to allow good cell division. So for productive mitosis, cell division, and nuclear processes, lamin A and the nuclear lamin are critically important. So I'm going to discuss some of the biochemical features behind how a lamin A mutation can cause HGPS. So lamin A, once it's produced in a precursor form, pre-lamin A. Once this protein is produced, there is a hydrophobic attachment called a farnesyl attachment, which is attached to the C-terminal of this protein. This farnesyl attachment I'm discussing because it's clinically relevant in that it confers a hydrophobic nature to this protein and allows it to stick to cell membranes. Normally, in the normal processing of this protein, this hydrophobic attachment is actually cleaved off. And this forms a mature lamin A and allows the mature lamin A protein to keep its normal conformation, its normal shape here in the nucleus. In HGPS, the gene mutation in exon 11 of this gene uncovers a cryptic splice site. And this novel cryptic splice site confers dysregulated or disordered processing of the protein such that this farnesyl attachment remains attached. So because this farnesyl attachment remains attached, the protein is now sticky and binds to different membranes. And as a result, the cell nuclear lamina then becomes disordered and unstructured. And this obviously hinders productive mitosis. And as a result of this dysregulated mitosis, the cells undergo premature molecular aging. So they undergo senescence, and they develop phenotypes that are consistent with disordered processing or dysfunctional processing of other pathways and proteins. So I'm just going to be discussing here the clinical phases of HGPS, and this is an arbitrary nomenclature. There's only 500 people in the world, and this is how, based on my review of the literature, I understand the disease. The disease can be categorized into early, intermediate, and late phases. I'm going to be discussing the early phases first. The average age of people afflicted with HGPS is about 15 years old, 14.6 years old. And so in the early phases of this condition, the clinical phenotypes are dominated by integumentary and musculoskeletal changes, as well as systemic conditions, renal and liver disease. This figure was actually taken from the original article that was in Lancet in 1886. So one of the most salient phenotypes for people who have HGPS is they have a very characteristic integumentary change that's characterized with dysmorphic faces. The patients have a beak-like nose with a small chin. Their facial features encompass a smaller proportion of their head, and the patients have a loss of subcutaneous fat. The patients also, by about 1 year of age, 100% of HGPS patients develop alopecia or complete hair loss. As a result of the loss of subcutaneous fat, the patients also have these venous telangiectasias. The patients also undergo a series of skin changes that are characteristic for advanced aging. The skin undergoes epidermal hyperplasia, increased relative water content. And so the skin takes on kind of a wrinkly appearance that we kind of associate with age individuals. So these patients develop a mild form of anasarca. So they develop dysmorphic faces, alopecia, anasarca, and along with this and musculoskeletal changes, the patients starting around 6 months to 1 year of life show significant growth divergence. So they fall 2 standard deviations below the mean and are within the 3rd to 5th percentiles of growth and maintain the 3rd to 5th percentile of growth throughout their lifespan. The growth that is achieved for these patients is defective. The patients develop severe osteopenia, osteoporosis, have decreased range of motion, develop pathologic fractures. And along with the musculoskeletal changes, one of the most salient is actually what occurs in the eye. These patients have deficits in the ocular muscles in the eye that govern accommodation and extraocular movement. They have visual impairment. The patients here, this was a case series that was actually released through Brown at Brown University, and they looked at the clinical phenotypes of these patients over time, and they noted that between 5 to 10 years of age, or 1 to 5 years of age, in the early phases of disease, the patients develop a liver and kidney dysfunction. They have a coagulopathy, and they develop transaminitis. on the liver side and on the renal side, they actually develop, a proportion of patients develop renal failure. And interestingly enough, they develop deficits in phosphorus and magnesium mishandling. And it's becoming appreciated in the aging field that actually phosphorus or hyperphosphatemia is actually a contributor to a lot of phenotypes associated with aging, including atherosclerosis. This brings us to the intermediate phase of the condition, which is of interest to us. Majority, or about 80% of patients with HGPS die of cardiovascular conditions. They're vasculopathic. In this intermediate phase, there's an immune and vascular endothelial cell dysfunction, which helps contribute to these late-stage vasculopathic features. So I'm going to be discussing some of these features here. In the aorta of these HGPS individuals, they show a characteristic histologic change. A normal vessel is composed of 3 layers: intima, media, and an adventitia. The intima is endothelial cells, and media is smooth muscle cells. The adventitia is support cells containing the vasa vasorum, and also fibrosis cells, and also support cells, adipose cells. Patients with HGPS develop a very thickened adventitia, and concomitantly, they have destruction of the smooth muscle cell layer. So with this increased change, with these histologic changes on an ultrastructural or electron microscopy level, there's increased vascular debris. As a result of this vascular debris, these patients have immune deficits. They have a very characteristic immune deficit in that they have an expansion of CD19 B cells. B cells are cells that make antibodies. So perhaps these individuals have increased B cells because of apoptotic or dead cells in the periphery, which allows expansion of this B cell population. The patients here start to develop vasculopathic features. These include a loss of vascular compliance or capacitance, elastic compliance of the vessel. So they develop both systolic and diastolic hypertension. And this is manifested echocardiographically as diastolic dysfunction. The patients have signs of LVH and valvular sclerosis of both the aortic and mitral valves. The patients also develop glucose and insulin intolerance, and a subset of patients develop frank diabetes. So this is the intermediate phase of the condition, and this ultimately leads to the late phase of the condition, which is vasculopathy. So these patients here develop very severe coronary calcifications. This is a case study here showing calcifications for this HGPS patient. Afflicts the left main, the LAD. This is also confirmed by IVUS. The cardiovascular phenotypes here with the vascular calcifications is not confined to the coronary tree, but also involves the cerebral vessels. These patients develop carotid stenoses as well as peripheral vascular disease. As you can see here in the histologic sections of this patient, they develop very, very severe vascular calcifications, eccentric lesions which contain macrophages. So these lesions in these patients very closely histologically and phenotypically mimic atheromas that are seen in aged individuals. And here, these patients here develop increased carotid ICA velocities. I mentioned they develop carotid stenoses, they develop strokes, and peripheral vascular disease. As mentioned before, about 80% of patients with HGPS die due to complications of cardiovascular disease. And these are some of the conditions that have been reported that have caused mortality in HGPS patients. These are very similar conditions that we see in aged populations in which we treat. So I'm just going to be discussing here a clinical trial in which HGPS was actually druggable. HGPS, as mentioned before, is caused by defective processing of a farnesyl attachment to this protein. Farnesyl is an attachment that's attached to the C-terminal end of this protein. And in HGPS patients, it's not cleaved off, so the protein becomes hydrophobic and sticky. Schering-Plough in the early 2000s developed a farnesyl transferase inhibitor. This is an enzyme here that mediates attachment to this farnesyl residue. And this drug was called lonafarnib. It was developed by Schering-Plough in the early 2000s prior to its merger with Merck. And there's, from what I gather, there's only been one published it was developed for the indication of solid tumor malignancies. There's one published abstract testing lonafarnib in the setting of non-small cell lung cancer. And because of its modest effects, Merck decided to actually license this drug to Eiger Pharmaceuticals in 2010. And Eiger Pharmaceuticals repositioned this drug for treatment of HGPS as well as hepatitis D. So in this trial here, this pivotal trial here, which is published in JAMA, HGPS patients were prospectively followed. This is a cohort study. There's 63 patients per arm. And as I mentioned, there's approximately 500 children in the world who carry this condition. So this is approximately 20 to 30% of the entire world population of HGPS who's enrolled in this study. And they were followed over a period of a median follow-up of 2.2 years with a primary endpoint of all-cause mortality. The cohorts were matched according to age, gender, and continent of residence. So the patients who received lonafarnib, you can see here, developed about 50% reduction in hazard ratio of all-cause mortality over a period of 2 years. So this really changed the field. And it's a really illustrative example of where a molecular basis can inform a therapy for a previously untreatable condition. And since that time, therapies for HGPS have focused on combination therapies. There's 2 trials, one trial that incorporates pravastatin and a bisphosphonate to improve functional status and weight gain in these patients. And also an ongoing trial incorporating lonafarnib with everolimus. Everolimus is an mTOR inhibitor, it's a part of stents, and this drug can stimulate a process called autophagy or self-eating. So all the cellular debris in the vasculature, it's thought that everolimus to kind of stimulate the body to kind of eat or clear that debris. And so these trials are ongoing. And what's also exciting is, as many of you heard, is gene editing technologies. I'll just briefly discuss here the base editor. There's a investigator at MIT, David Liu, and it has a base adenine editor that can infect about 30% of cells and has increased the lifespan of mice in preclinical studies by 30 to 50%. And because HGPS is a rare orphan condition, the thought is to gain FDA approval here, it might be able to be repositioned to treat aged individuals or age phenotypes in the normal population. And this is just that study here, which I'm not going to go through here, but you can kind of see the dramatic increase in the mortality of these mice. And it should be mentioned here, well, why we study, this ultra rare condition that affects about 500 patients in the world. And this progerin or this defective lamin A protein actually gets accumulated in normal individuals. So we as individuals in an age-dependent fashion accumulate this progerin or the defective lamin A protein. So it's possible that perhaps the therapies for HGPS could potentially affect lifespan for individuals without this condition. And as I mentioned before, HGPS mimics a lot of the aging phenotypes, including the cardiovascular phenotypes. And so it's an interest. It's a field of strong interest in the molecular biology world. So I'm going to back up here to talk about a gene. It's called Z521, which our lab studies, which recapitulates a lot of these phenotypes for HGPS. My lab typically studies solid organ transplantation, and we study process allograft vasculopathy, which is an immune process where the vessels in a T-cell mediated fashion kind of close in the transplanted tissue allograft. It limits the longevity of these grafts to approximately 10 years. Complement are a set of immune proteins that are critically involved in allograft vasculopathy. It's prognostic for, diagnostic for, and a therapeutic target for allograft vasculopathy and transplant rejection. Complement proteins, there are 9 proteins. They're produced in the liver and they circulate as inactive proteins. They get targeted towards the donor vasculature, the donor endothelial cells by antibodies. So during antibody-mediated rejection, complement gets activated on endothelial cells. As a result of the complement activation, the complement proteins self-assemble and they form this pore. It's a pore-like structure called the membrane attack complex. And it was thought initially that this pore could cause tissue inflammation by causing cells to die by osmotic lysis. These were slides taken from the Yale pathology here, the Gil Meckel. And you can see here, there are 2 salient observations that could be made. The first is that complement deposition during transplant rejection principally affects only endothelial cells. So only endothelial cells are affected by complement. And the second is that the vascular tree, or the vascular architecture is actually preserved in these patients. So actually, even though they have MAC pores, these pore-like structures on the surface, the cells don't actually die. And so this raised the speculation that membrane attack complexes could signal. They actually function as a de novo receptor that actually can initiate signaling. And this is what my lab studies is MAC signaling. We have a protocol which we can induce MAC deposition on endothelial cells using a human transplant serum. The details I'm not going to go over here. This is a molecular basis of that. Patients typically, because of the intrinsic risk of complement on the tissue allograft, they undergo screening called panel reactive antibody screening. And we recognize that there's a cohort of patients that have a very, very high PRA percentage. So these patients have a high titer of non-self-binding alloantibodies. And we use this as an investigational tool to intentionally cause MAC deposition on human endothelial cells in vitro and also in humanized mouse models. And you can see here that what we're trying to advance, or what we're trying to show, is that MAC as a signaling receptor, the way that it signals is that it creates an internal compartment of these signaling endosomes, so these internal vesicles. And these internal vesicles actually, they act as like a sanctuary city. They actually sequester pro-inflammatory molecules. And then when they sequester the pro-inflammatory molecules, the half-life of these molecules is increased. So rather than being degraded, the molecules are propagated, and this causes tissue inflammation. So those are the two hypotheses we've investigated. MAC is a receptor. MAC causes signaling endosomes that sequester inflammatory proteins to increase their half-life. As an extension of that hypothesis, we reason that these signaling endosomes, or these vesicles here, are enriched in signal activating or inflammatory proteins. So we isolated these signaling endosomes, and that's how we discovered Z521. I'm not going to show the painful molecular details, which that's the focus of my life every day, the painful molecular details. I'm not going to go through those today, but I will say just briefly what Z521 is and how we got into aging. So Z521 has only been studied by one group. Very literally, these are the 4 papers that have been published previously by Z521. This group is no longer active. The last paper here is in 2013. So unfortunately, and they were very, very good. They were from the University of Tokyo. I kind of wish in a way that they still studied the disease. I think we'd know a lot more about the molecule if they were still there. But we're the only group in the world that studies this gene. Not much is known about it. It is implicated in cell motility and tumor adhesion. We found that Z521 was involved in upregulation of inflammatory genes. In a humanized mouse model, we found that a drug called miltefosine, which is an FDA-approved drug for breast cancer, when repositioned to treat mice in a humanized mouse model here, we can treat allograft vasculopathy. In this model here, we get human coronary arteries from the operating room. We get them from George Toledes and Arnar Gearson, Patrick Davis, and we implant these arteries in the descending aorta of these immunodeficient mice. We then engraft the mice with human lymphocytes, and then we study this condition of allograft vasculopathy. It's an in vivo humanized model. Miltefosine can inhibit allograft vasculopathy by inhibiting Z521. So Z521 causes endothelial cell dysfunction. Inhibiting this gene blocks endothelial cell dysfunction. Z521 was found in the endothelial cells of at least 5 different patient cohorts. These are biopsies from patients with rheumatoid arthritis, osteoarthritis, cardiac allograft vasculopathy. And what's not shown here is lupus nephritis and kidney biopsies for patients with antibody-mediated rejection. So these Z521 is involved in endothelial cell dysfunction. It's expressed in humans, and it's a very highly conserved and ancient protein. This is a phylogenetic analysis of the gene. This gene here, Z521, is estimated to be between 500 to 1,000 million years old, so 1 billion years old. It's found in sea anemones, which evolutionary biologists use as a landmark for evolutionary age. And it's highly conserved in all higher order primates. And usually when genes are highly conserved and very ancient or old, usually they have a very fundamental biological function. That means the function of this protein is likely very important. So it's in the endothelial cells, causes endothelial cell dysfunction. It's likely a very fundamental molecule. So we investigated this gene in the setting of aging. We developed a Z521 knockout mice. Again, the molecular details are not significant here. What I will say is once we developed the construct, based on our study, we knew exon 3 was required for the function of this gene. We bred the mice in such a way such that when the mice were given a drug tamoxifen, Z521 is knocked out specifically in endothelial cells, and then the endothelial cells turn green so that we can isolate the cells for downstream studies. So when we isolate the endothelial cells after giving the mice tamoxifen at an RNA and protein level, the gene was knocked out, and looking at the blood vessels, the endothelial cells are green. So this satisfied the fundamental tenets of our breeding strategy. And we subsequently set out on a series of kind of observational or survey to see how Z521 could affect aging. We found that similar to patients with HUTCHINSON-GILFORD progeria syndrome, the mice develop increased water weight that affects all organs. So this is a dye called Evans blue. The mice are injected with this fluorescent dye, Evans blue, which binds albumin. So wherever albumin goes, that's where the dye goes, and that's why the organs turn blue. This implies that there's a breakdown of vascular integrity, and there's extravasation of fluid into the interstitium, implying increased water weight or anasarca, which HGPS patients have. These mice show severe growth retardation. They're about 25% less in weight compared to their wild type litter mates, again, mimicking HGPS. The mice develop signs of osteoporosis and osteopenia. They have decreased bone mineral density. The mice have a very profound hypoperfusion of the retina. These are retinal vessels here. And as a result of this hyperperfusion of the retina, these mice likely have a visual impairment. The mice also develop renal dysfunction. You see here the Bowman space in the glomeruli is increased. There's interstitial edema, some cystic changes. Ultrastructural studies show that these are podocytes, and they undergo effacement. Podocytes are support cells in the glomeruli that regulate perm selectivity or ultrafiltration. Loss of podocyte function or effacement of podocytes is shown here as a marker of renal cell injury. These mice develop hyperkalemic renal failure. These are the potassiums in these mice. A normal creatinine in mice is 0.12. So these mice develop a hyperkalemic renal failure. They develop proteinuria. So they have increased, this is protein creatinine rate, albumin creatinine ratio. They have increased proteinuria and they have hypoalbuminemia. So these mice actually develop a lot of features of nephrotic syndrome. They don't have nephrotic range proteinuria, but they have edema, proteinuria, hypoalbuminemia, and probably the mice may have hyperlipidemia. And I'll show why later. This is a liver slide here. And this is why I think the mice have hyperlipidemia. They have signs of liver dysfunction, signs of extramedullary hematopoiesis, hepatocellular degeneration. The pathologist here also told me there's Mallory bodies. So there's signs of liver injury, kidney injury, musculoskeletal changes, integumentary changes. These all mimic features of HGPS. What is not found in HGPS, which is found in the mice, is they develop COPD-like changes with increased or widening of the air spaces. The mice also develop severe splenomegaly, and very similar to the HGPS patients, they develop the B cell deficit. They have an increased number of B cells, but not an increased number of T cells. So this phenotype is very similar to what's found in HGPS patients. The mice also develop hypertension. They have signs of histologic signs and clinical signs of hypertension. There's hyalinosis of these vessels, onion skinning of the vessels, kind of compatible with hypertensive changes. And this occurs in all organs which were found. I didn't label them here. This is the lung, the brain, the liver, and then the heart. You can see here also that these little punctate changes here, there's perivascular edema in the heart. The mice are concomitant with hypertension develop diastolic dysfunction. They have increased LA size, increased intraventricular relaxation time. They develop increased velocities through the aortic valve, suggestive of aortic sclerosis, which is shown here. The mice also develop glucose intolerance. There's a cohort of mice which have frank diabetes, which was defined as a random glucose greater than 200. So a lot of these vasculopathic features in the Z521 mice kind of mimic what we think in HGPS. We haven't done any studies yet to define vasculopathy or coronary artery disease, strokes, peripheral vascular disease, but these studies are ongoing. This is just a summary of the phenotypes that we have so far. As you can see, HGPS and Z521 have a lot of features in common there. I think this model here will be useful in order to study the molecular basis behind aging. Again, in an effort to try to improve longevity of patients rather than secondary prevention of the disease, we can consider senescence or aging as the first hit or the primary druggable hit, which might prevent these downstream syndromes. So preventing senescence might be able to prevent 4 or 5 different cardiovascular conditions. And this is just a hypothesis. But suffice it to say that just to affirm the clinical relevance here, this is the ICD-9 code of the most costly medical conditions here in the United States. HGPS and the Z521 mice recapitulates here in red, most of them. So I think this is a very clinically relevant thing that we would like to interrogate further. It's a significant divergence from what my lab typically studies. These are the conclusions here. And these are the acknowledgements of the people here and the funding agencies and their collaborators. I'm part of the VBT Institute here. And my mentors here, Jordan Pober and William Sessa. I do my clinical work at the VA. I'd also like to acknowledge here not shown Bob Sufer and Steve Fow, and also Mike Coswell, who helped to, who really was supportive of my research program. I'll be happy to take any questions here today.
Thanks, Stan, for taking the time to talk us through, really interesting talk. I think I can get started with the question. So one of my key, thoughts that came through was there's a lot of overlap in, as you mentioned, different mechanistic endpoints which lead to vascular dysfunction, liver dysfunction, kidney dysfunction, recapitulating aging, advanced aging in HGPS. But does the fact that a monogenic sort of pathway leads to that aging mechanism change does it feel like that particular pathway that particular disorder has, which is accelerated aging, would apply to normal physiologic aging?
That's a really good question, and this is the home run kind of thing that people want to show with these genetic diseases. It's the monogenic condition that can cause these conditions. It is possible, and I just discussed just the genetic basis here, but there are other contributors of aging. It is likely, it is multifactorial, that increased oxidative or metabolomic products, environmental factors, risk factors, diet, other obesity, other conditions that are modifiable may very well have a modified basis on the initial genetic foundation. We haven't interrogated those. Those conditions are kind of challenging to interrogate. Well, I say just as an aside, a way to distinguish between these different risk factors is actually in the skin. In the skin here, there's 2 processes. One is in the skin, there's a basal cell layer, which basally proliferates. And as the skin cells quantify, they rise to the surface and kind of slough off. So the epidermis is continually proliferating. So that rate of proliferation can be measured as a sign of senescence. Now in the skin, it's exposed to sun layers. And so the penetration of the skin into the dermal layers where those basal cells proliferate is about 83%. What people use as a comparator to that is actually skin underneath in the nail bed, and that the sun only penetrates around 17% into these basal cell layers, but yet in the nail layers, the skin still proliferates. And so they compare the proliferative replicative capacity of cells within the nail bed to cells in the skin to kind of distinguish between these two. And these models here are just in order to try to define molecular basis of senescence. No one really knows if progerin or these molecular byproducts are toxic. That's a very good question. I think it's multifactorial. I think the genetics is a foundation.
I have a couple questions in the chat, please. Okay, so we have Jeff and Barry. So Jeff, do you want to take it on?
Sure. Dan, that was an outstanding talk and terrific work. Thank you for that presentation. I'm trying to wrap my head a little bit around the connection between Z521 and HGPS. It looks like the knockout mice do recapitulate the syndrome in a major way. but you haven't really described to us how or why that occurs, given the lamin A, hyperfarnesylation, if you will, or farnesylation defect. So I'm wondering if you can make that connection. A lot of small G proteins are prenylated, so I don't know if RAB5 is farnesylated, and there's any connection there between the signaling endosome that you've so nicely described, Z521, and HGPS, just as a question.
Yes, no, that's a good question. I think that even though the clinical phenotypes might mimic each other, the molecular basis may not necessarily. We are interrogating some of these molecular changes which may underlie senescence. And what we have found is that loss of Z520 profoundly affects cell replication, and that there's evidence of double-stranded DNA breaks in these mice, and that there's evidence of increased tumor suppressor genes, including P53 and P21. So what we think is that Z521 may be affecting the overall health of the DNA, whether or not that's due to defective replication or just due to oxidative injury of just non-dividing cells, whether or not cell proliferation is required for this process, for this senescent process, is unclear at this time. The mice were just newly generated, and I should just mention that with each subsequent cross, I just kind of gloss over the details. We have 3 crosses that were made. So each cross is about 6 months in time. Generation of the construct was about a year in time, when with COVID, supply chain shortages and the rest. It's been a very long road to generating these mice. And we do look forward to try to phenotyping these mice using the molecular cores through Dr. Sanusis here, through the O'Brien Kidney Center, and also through the Liver Cancer Center, the Liver Center, the Cancer Center, and also through endocrine to kind of determine the molecular basis of how Z521 might work. Because it's multi-systemic or multi-organ, I suspect that the molecular basis will vary according to organ.
But
since we're cardiologists, I think we will be embarking on studies to study the role in coronary artery disease.
Okay, great, thank you.
Yeah, that's a good question.
Yes, Dan, great talk. I grew up in a different era where we thought of aging in association with amyloid protein. Can you think of any potential interaction between the genetic defects you've shown and the processing or the development of amyloid protein, looking for a unifying mechanism for a number of other events?
Yeah, we have looked only in the setting of amyloid protein and setting of Alzheimer's. I didn't show here today, but the vessels in the brain show beta-pleated sheets or Congo red staining in the vessels, implying they have amyloid angiopathy or cerebral amyloid angiopathy. We don't see signs of tau. We don't see signs of neurofibrillary tangles or any signs of neurodegeneration. It should be mentioned that these knockout mice here, all the phenotypes which were derived from these knockout here were loss of the gene only in endothelial cells. So this just kind of highlights how critical the endothelial cell is just to the biology. I wouldn't have typically associated with osteopenia, osteoporosis with endothelial cells, nor liver damage with endothelial cells. But we haven't looked at, in answer to your question, we haven't looked at amyloid protein aside from that. That's all that I can say about the amyloid protein. That's a really interesting thing. We do know that with knockdown of Z521, the APP gene or presenilin gene, which encodes the amyloid precursor protein, is unchanged. And that's all that I can say about it.
Thank you.
Hey Dan, if I may ask a question. So first, congratulations. I think you've made a real discovery.
Thank you. Yeah.
So you must have done some RNA-seq to look at gene signatures in endothelial cells, and I wonder, sort of which signatures popped out.
Oh, well, as you know, the RNA-seq gene data sets are only as informative as kind of the bioinformatics that kind of underlie them: cell proliferation, cell growth, DNA damage pathways, and there's all sorts of other cancer pathways, but DNA damage and cell growth popped out as salient pathways. And I don't know, as I mentioned in answer to Jeff's question, whether or not Z520, it's a protein that's mostly expressed on an endosome. So to assign a nuclear function to that, we haven't kind of gotten that far on a molecular basis yet. So we don't have a good molecular understanding of how it works. And I think, yeah, the RNA-seq data certainly may help, or single-cell RNA-seq may help once we have the global knockouts as well. Yeah, that's a good question.
Dan, can I raise a point?
Yeah.
Hello?
Yeah, we can hear you.
The amyloid question is a good question as well. That's interesting. That's really interesting.
All right, please go.
Yeah.
So I have, Dan, a question for you, and maybe a point actually, and that is the link that Jeff asked. First of all, congratulations, beautiful work, amazing phenotyping of the mouse. This was great job, and you have discovered, as others said, Martin said, etc., something very, very novel. Now, the link between the two diseases actually can come probably from a little bit of understanding the genetics of these lamin A mutations. As you know, other mutations in lamin A cause familial partial lipodystrophy, a disease very similar to progerin, in that actually patients develop atherosclerosis, they die from atherosclerosis, they have diabetes, and they have what is called loss of fat in subcutaneous tissue. Now, this actually is treated, in fact, actually with transplanting some fat to the buttock in this patient, partial lipodystrophy. Now, I wonder actually there is at least similar etiology of the diabetes that you have in this mice with practically its form of lipodystrophy, that it causes lack of development of the lipid droplet, and then lipotoxicity that can actually cause organ damage. And as you know, the Z5 is involved actually in kind of aggregation of some lipid droplets in the endoplasmic reticulum, and has some function there through, also itinerary of the lipid droplets and lipids inside the cell. So my question to you, is these mouse also have something like lack of adipose tissue subcutaneously or not? Although you have done it more endothelial knockout, but it still could be. And second is that the etiology of diabetes is quite intriguing. Is that by loss of insulin secretion or insulin resistance? That would be easily addressable with some doing an oral glucose, intraperitoneal glucose tolerance test, and maybe even a clamp study.
Yeah, thank you, Arya, for the question. Congratulations to you too with the Berliner professorship. I just wanted to say that we don't see steatosis in the liver there. We haven't looked specifically on whether or how Z520 affects endocytic or secretory rates. I suspect that it will be a critical part of why this disease or why these clinical manifestations occur.
Is it a loss of subcutaneous fat in these mice too, or no? Like progeria?
We haven't phenotyped that yet, but I suspect that there will be a difference or a loss in subcutaneous fat is what I suspect. But we haven't actually looked at that yet. And we would try to connect with the Yale pediatric group here to try to find patients that might carry the Z520 mutation, because as I mentioned here, as strong as the phenotypes here, this is only a knockout, only in endothelial cells, and these mice are 8 weeks old, which is equivalent to somebody, a human between 30 to 40 years old. It's not a very old mouse. So these phenotypes develop spontaneously. It involves only one cell type. And so a global loss of this would be expected to have a very severe phenotype. And there must be some cohort of patients that might be walking around with this kind of condition that's unclear or unknown. We haven't had any, we're in the process of trying to find, but we haven't found a source or maybe a proband of patients that might carry this mutation. Yeah. Hey, Kat, how you doing?
That's beautiful work, Dan.
Thank you.
With all these other genes that you see in progeria, do you see any changes in the nuclear morphology, the nuclear envelope, or chromatin organization with your ZF21 knockout?
Yeah, so we haven't stained for LAMIN, but for DAPI or just a nuclear stain, it doesn't appear that the nuclei are misshapen. They are smaller. They are smaller, but they don't appear misshapen. And this is a new field entirely for me, you know, study here to here, you know, and with transplant there. So this field is new for me as well. And I think there are different ways or ways to approach it at kind of an executive or PI level, is that maybe there's a unifying, and I think Dr. Bender alluded to this, where there's a unifying molecular mechanism that kind of underlies everything. That there's one thing that causes it all. And it is possible that's the case. I suspect that in different organs, Z521 may have differential functions. Because different organs have different replicative capacities, the liver proliferates more than the brain. As an example, the junctional proteins in the brain and the vasculature in the brain is different than in the kidney, which is much more leaky and more permissive for filtration and urine production. So I think that the roles in each of these organs will be different, is what I suspect. But that's a good question. We haven't looked.
And nice work. It's been fun for me to follow this along over the last several years. Congratulations.
Thank you. Thank you.
So my question relates to a comment you've made on several occasions here, talk about the potential durability of these. Really, question around what you see as the future for your laboratory as we move in a direction towards that. So is particularly the D521, are you able to overexpress or change it in some way that you can, and then move to the liver so that you can use animal liver controls as you look at these actions? I think I suspect there's multiple companies would love to figure out how this works with the skin. But you know, just your thoughts about the next series of years of experiments in this field that you're developing.
Yeah, I think there's a lot of pharmaceutical interest in cosmetic things. I think alopecia and reversal of skin changes is really a big molecular focus, and delivery of genetic. So I think the future here will require genetic modification, and I think as it's becoming more and more appreciated that. Especially with the recent transplants, the first xenotransplants for kidney to human, heart to human, lots of conditions there that involve genetic manipulation. You only need to modify a small percentage of cells in order to get a phenotype in the skin. So for example, there's a study showing in Nature Communications, 20 to 30% gene modification is sufficient to restore these skin phenotypes. As I mentioned here, the paper involving CRISPR-Cas9, this gene therapy here involving AAV9, it's an adeno-associated virus, it only affected about 30% of it. So only one treatment. And the treatment can be done postnatally or can be done as a secondary preventive measure. And I think a genetic manipulation, as it becomes more and more accepted, will be the way. As far as where my lab is going, yeah, I wish I knew, you know, it's just been such a very unusual. I never would have imagined that I would have worked on or come here. So I'm not entirely certain. I want to study the interface between the immune cells and vasculature, which we have funding for. And this is where I think I will go, but it's hard to say. We've actually identified another gene. It's probably outside the scope here that we'll probably be working on. It's called WDFY1. It's another Rab5 effector like Z521, something that we might interrogate in the future. Yeah, thank you very much. I wanted to thank everyone for giving me the opportunity to speak here today. Yeah, appreciate
Thank you.
Before everyone leaves, I just want to remind everyone that our own Rohan Kara is giving medical grand rounds on Thursday morning. I think it's a hybrid environment, so those who are around can come here. But please also appreciate if you can also attend, if you can't in person, hybrid and medical grand rounds. Remember, starts at 8:30 on Thursday. Thanks, everyone.
Thanks, Eric. Thanks so much. Thanks, everyone.